Okay, so this lecture is about genetics. And genetics, like we mentioned before, is the study of heredity. So we are going to look at some examples of what we know about heredity and how genes and traits are inherited. We also will look at some of this hands-on in the lab. So you'll read about this and then we'll see it in lab too. Okay, so Gregor Mendel is usually attributed to a lot of what we know about genetics. He's known as the father of genetics. He began a research project in the mid-1800s about inheritance patterns. So he was working in the garden a lot, and he would see how different plants would inherit different traits. And he did thousands, it says 30,000 pea plants. He did thousands of experiments and trials to try to understand how different traits were inherited, and he was using pea plants. So what he was looking at in pea plants, we have also then expanded to other organisms and learned about inheritance and heredity in all different areas. So he demonstrated that traits, which is different, we'll define traits here in a minute, but they're transmitted from parent to offspring in specific patterns. So when he was looking at pea plants, he was looking at different traits like size, short, tall, flower color, the size of the peas, the size of the pods. So obviously this is pretty simple in a system like a plant, and it gets much more complex when we're talking about Homo sapiens or different organisms. But everything that we learn about genetics in this more simple system, we can kind of apply to more complex systems. So this species of pea plant that Mendel was using naturally self-fertilizes pollen and the ova of the same flower. So this flower has pollen and ova within the same flower, and the flower petals remain sealed until pollination is complete to prevent pollination from other plants. So plants produce offspring that often look like the parent plant. So first he saw mating two true breeding individuals so that they have different traits. And a trait is a physical variation or heritable, which just means it can be inherited characteristic. So in this case, it's something physical that can be observed, like the plant color, the size, the shape of the pea pods, that sort of thing. So plants used in the first generation were called P or parental generation, and then offspring of the parental generation were called F1 or first filial generation. So this would be like the offspring. And then they would examine the exhibited traits. So the second generation would be crossing two of the F1 parents to then produce F2. So this would be like a person and then their child and then their grandchild if we were looking at it that way. And they saw that the F2 generation exhibited 705 violet flower plants and 224 white flower plants. So this resulted in what they called a ratio of exhibited traits. And then they would repeat the process with subsequent generations to see what kind of flowering pattern they got. So these were some other traits they looked at. So they looked at seed color, seed shape, flower color, pod shape, and pod color. So he collected all kinds of data about these different traits and how the different plants were being crossed. And what he found was this ratio in traits. So obviously these numbers are not exact, but they all were about 3 to 1, 2.98 to 1, 3.01 to 1, 2.95 to 1, 2.82 to 1. So all of these numbers are kind of close to 3 to 1. So when he observed that, he determined that characteristics could be expressed, could be divided into two categories, what are called expressed and latent traits. Expressed traits are also called dominant traits, and latent traits are called recessive traits. Recessive traits become latent or disappear in the offspring, so they're not observed, and the expressed traits are usually observed. So, the reason that this is so is because in a pair of homologous chromosomes, one of them is inherited from the female parent, the other from the male parent, and the gene is located in the same place on the chromosome. So alleles are different forms of the same gene. So, for example, if you have a mom and a dad, and this is the gene for eye color, you would have two alleles for eye color. You would have one allele from your mom and one allele for your dad. So a pair of alleles may not be identical, and alleles that are represented, so it might be a dominant and recessive, so then the offspring is some combination of the alleles of the parents. So a gene or a segment of DNA that contains the code to build or regulate a protein. So we have two copies of these, so we have two alleles to form a gene. So in this case, we see a dominant allele, which they are representing with a capital Y, and a recessive allele, which they are representing with a lowercase y. So a dominant trait, if you have even one copy of the dominant allele, then you'll express the dominant trait, brown seed color, and the only way you would see in a green color is if you had two copies of the recessive allele. So if you're big Y, then you have a brown seed color. If you're big Y, even though you have an allele for both brown and green, because brown is dominant, you would have a brown seed color. And then if you have two recessive alleles, little y, that's the only time you would see a green seed color. So genotype are the alleles that are present. So there's three possibilities for genotype. There's homozygous dominant, which would be big Y, homozygous recessive, which would be little y, and then heterozygous, which would be big Y, little y. So those are the three alleles that are present. The genotype, heterozygous, having two different alleles, the dominant will be expressed. Phenotype is what is observed. So genotype is what the actual alleles are. Phenotype is what is observed. So the phenotype is dominant or recessive. So if you have two seeds that you're crossing and you have big Y, little y, you're going to get 100%, all of these are going to be yellow hybrids because they all are going to be heterozygous, big Y, little y. And we just said that heterozygous, big Y, little y is going to express the dominant trait. So we'll see examples of each of these when we are doing the genetics lab. So this is another example. Now if we cross that F2 generation, if we cross two heterozygotes, if we cross big Y, little y with big Y, little y, we would end up seeing a genotype that would be 25% big Y, 50% big Y, little y, 25% little y. But the phenotype is going to show us 75% that are dominant and only 25% that are recessive. So 3 genotypes, but 2 phenotypes. So a monohybrid cross is when you are crossing two different true breeding parents that differ only by one characteristic. So the experiment that was done here is that if we have an F1 offspring that has the phenotype of one parent, so violet flower plants, and then the F2 offspring had a 3-to-1 phenotypic ratio, 3 violet flower plants for 1 white flower plant. So we'll do an example like this. You could go ahead and give this a try and see what you might get when you cross these two parents, see what genotype and phenotype you would get for offspring. Another example would be to demonstrate, this would be two heterozygotes. So they both are expressing the dominant color because they have one copy of the dominant allele. They're heterozygous, meaning they have one dominant and one recessive, but what is expressed is the dominant. And my little picture in the video is kind of blocking it, but they all would end up being, when you are crossing big A, little a with big A, little a, you would end up with, you'll see this on the lecture, I can't move my little video, but you would have big A, and then you would have big A, little a, and then little a. So it would be 75% is going to be expressing the dominant trait and 25% the recessive. So 50% heterozygous, 25% homozygous dominant, and 25% homozygous recessive. So this is that 3-to-1 ratio that Mendel observed when he was looking at all of the different traits. Here is another example, crossing two heterozygotes, showing what we just showed on the previous slide. So you would end up with 3 purple flowers and 1 white flower. This one, looking at monohybrid crosses, so crossing a homozygous dominant with a homozygous recessive, you would end up with 4 heterozygotes, which all would express the dominant trait. So if you cross a homozygous recessive, which is white, with a homozygous dominant, which is purple, you would end up with 4 purple flowers, and they all would be heterozygous, but expressing the dominant trait. Here's one more example, and you can do this on your own. We also will see examples of this in the lab. This is crossing seed colors. So this would be heterozygous and homozygous dominant and homozygous dominant. And also you could show what the different colors are and then what the phenotype ratios are. And then one last example for you to go through. And also these are in the previous slides and in the textbook. So take a few minutes, go through this, see what you think. We'll talk about this more in class and we'll cover it in lab. And that's where I'll leave it for genetics.